Hello, hello, biology. Okay, video is for those of you who were not here this week. I'm going to do I'm going to do one video kind of outlining the whole week. And then next week, I'll probably do one per day since I was not very on top of things this week, and I apologize for that. So, Monday this week was a day to work on stuff while we were yellow. This day, Tuesday, is the first day where we actually started our new unit. So this was the bell work, and then this assignment. If you have not done this, this closed today. I'm recording this on Friday. This closed today. I'm just going to exempt you from this if you haven't been here, because that's not your fault, and I apologize again for that. So we are doing DNA and protein synthesis in this unit, and I'm going to try to speed along since I was told by several people that they prefer Miss Stauffer's style of note-taking. So DNA stands for deoxyribonucleic acid. Nucleic acid is one of the 4 macromolecules we talked about back in Unit 3, I think. Deoxyribose is the sugar base of that, and that's why it's called deoxyribonucleic acid, because it is a nucleic acid with the base, or with the backbone, deoxyribose. And that's what contains your hereditary or genetic information, and it is a double helix, frequently described as a ladder that has been twisted. So the rails, the sides of the ladder, are composed of 2 things, and then the rungs of the ladder are composed of something else. So this would be a nucleotide. Okay, so the phosphate group binds up here to another deoxyribose. It would bond like this. Be another pentose or another phosphate here. Bonds to the sugar, and then the sugar has a base, and then there is a mirror image of this on the other side with another base, then a pentose sugar, and then phosphate and a phosphate attached to the bottom. But this is the monomer. This whole thing is the monomer for deoxyribose. And for ribose, except that, the monomer for DNA, except that this sugar is different in RNA. Which I'm sure wasn't confusing at all. Man, I butchered that. Okay, so DNA has 4 kinds of base pairs. So each of those 3 parts has some variance in it. So that's what we're going to look at next. There are 4 different things that can be one of the bases, or the rungs that pair up. They pair up for the rungs of the ladder. Adenine, guanine, cytosine, and thymine, or A, G, C, and T, are the way that they pair up. So adenine cannot bind to guanine. So if we go back here, if this is adenine, then the next base over can't be guanine or cytosine. It has to be thymine, because those are the pair. If this is guanine, then this has to be cytosine. And then if you are a Highway to Hell fan, here is a way that some people remember it. The way my 3rd hour came up with this week was at group chat, which has worked for several people to help keep it straight. So at group chat for the pairs. If that works for you, awesome. If it doesn't, we need to come up with something that does, because this is something you have to know for this unit. You have to keep these straight. Okay, and then the phosphate, that's going to be your phosphate group. It's one of the 2 components that forms the rails or the sides of the ladder, along with your pentose sugar, which in the case of DNA is deoxyribose. So here's a diagram where your rung here is made up of guanine and cytosine. The rung below it is adenine and thymine. guanine and cytosine again. And as you move down, you're going to see that repeating. Now, there is a pattern here, but that pattern is not regular or consistent in any way. We'll talk about how that works later. Okay, the bases are held together with hydrogen bonds. That's what keeps these from drifting apart. And then your sugar and your phosphate group, so sugar binds to the base, and then up to the next phosphate group binds to the sugar, etc., etc. Those form the rails of your ladder, the sides. Here's another two-dimensional diagram again. So you can see our nucleotide. We have our base, our sugar, and our phosphate. The phosphate binds to the next sugar of the next nucleotide. This phosphate binds to the next nucleotide and the next nucleotide. And you can see that each base has to have a certain thing on the other side. Okay, we represent this frequently with an arrow and a curve to show that thiamine won't bind with guanine or cytosine. It just doesn't fit together. Now, chemically, this is a little more complicated than that, but that's good enough for this class. So nucleotide, a little bit of a throwback for you. Back to nucleic acids. Nucleotide is the base unit of DNA, made up of one base, one phosphate, and one deoxyribose sugar. A gene is the information contained in DNA. So DNA is a big, long sequence of millions and millions of base pairs. Okay? Within those millions of base pairs is a section of what could be anywhere from 12 to 1,000 bases that makes up a gene. The gene is the sequence of DNA that codes for a protein. Okay? Now, more complicated proteins need longer, I'm pointing in the wrong spot, need longer genes because a more complicated protein is going to require more information to describe it. But all proteins can be described using sequences of these four letters. Okay, to give you an idea of how tightly bound up or how long your DNA might be, if this is a chromosome, you'd have to pull out a thread and uncoil it, and then uncoil it again, and then uncoil it again, and then you'd actually start to see your DNA. Now, an individual gene on DNA is going to start with an exon. The intron is the actual information, and then an exon is going to be the stop codon. This is called the start codon. That's something we'll talk about next week. Okay, all DNA is found in the nucleus for a eukaryotic cell. For a prokaryotic cell, there is no nucleus. It's just kind of floating around in the cytoplasm. Chromosome is what you get when DNA has been tightly coiled up. Okay, and chromosomes are going to be collections of lots and lots of genes that have been put together. Oh, and then humans, 46 chromosomes, 23 from mom, 23 from dad. Here's another example of how tightly coiled it is. Okay, this is the crash course video that is linked below this video. And he's going to go through and kind of walk through DNA, and he's going to talk about RNA and hit a little bit on some of that and what's going on. The assignment at the end of the day, the end of class this day, was to answer these 4 questions after watching this video. So before you go on, I recommend that you follow the link, watch this video, and then come back and try to answer these questions. Okay, so that was Tuesday. Wednesday, we didn't do nearly as many notes, so this should go pretty quick. This was the bell work. And then we started it by going through those 4 questions. So these were the answers for those questions. Then my 3rd hour did not finish this video. So we finished this video on Wednesday. My 7th hour got through the whole thing that day. Because of some variance that was going on there. Okay, and then the rest of the hour on Wednesday was spent working on worksheet 1, which was due on Friday. And if you haven't done that, I'm going to open that back up for you so that you can go in and work on that. And that was pretty much it. Now, this says due Thursday. I extended that through Friday because we spent Thursday and Friday doing labs in class with worksheets to go along with that. So we had a little more work this week than I intended. So I did extend this out. So this ended up being due Friday, and I will open that back up. If you send me an email, let me know that you're ready to work on it, and I'll open it back up for you. That is pretty much it for Wednesday. Which brings us to Thursday. So Thursday, we did a little lab activity where the students built a little DNA molecule using some kits that Miss Stauffer got. You're going to have to come in and actually do this at some point. This isn't something we can do virtually. So at some point before the 8th of January, you're going to need to make arrangements to come in. Could be after school, could be before school, and actually work on this. After the 8th, the semester's going to end, and they're going to close the classes. So you need to get with me. Let me know when you're going to do that, because we also did a lab. Oh, and there were... worksheet. There was a worksheet on that. We didn't do that. Ignore that page. Ignore that slide. This was Friday. Friday we did another lab where we actually extracted DNA from strawberries. And again, you can't do that at home. You're going to have to come here and do that part. So again, let me know, because I'm going to have to go buy new strawberries, because those ones, they're rotten, or they will be probably pretty quick. I doubt they'll make it through the weekend. So you're going to have to let me know when you're going to come in so that I can buy new strawberries for you to use. So yeah, and then that was pretty much all we did on Friday. Today? Friday? That was pretty much all we did today. Then we worked on worksheet, whatever the strawberry lab was, the DNA extraction worksheet, and they answered some questions at the end of that. And that's pretty much it for this week. So that's where we're going to end this video, about 13 and a half minutes long. I'm going to go ahead and stop so this doesn't get long. Hope you all are having a great week.